I'm Jamie, and with me today is my co-host, Alex. We're diving into some fascinating new research about cancer treatment strategies.
That's right, Jamie. Today we're exploring a really intriguing study about a potential breakthrough in cancer treatment. It's all about making chemotherapy drugs called alkylating agents work better by targeting something called the G2M checkpoint.
This study was just published in Neuro-Oncology, and it's pretty fascinating. The lead author is Fengchao Lang, along with a team of researchers including James Cornwell, Karambir Kaur, and several others from the Neuro-Oncology branch at the National Cancer Institute.
So, Jamie, before we dive into the specifics, maybe we should explain what alkylating agents are for our listeners who might not be familiar with them.
Absolutely. Alkylating agents are a class of chemotherapy drugs that work by damaging the DNA in cancer cells. They've been used for decades to treat various types of cancer, including brain tumors like glioblastoma, as well as lymphoma, leukemia, and cancers of the ovary, lung, and bladder.
Right, and one of the most well-known alkylating agents is temozolomide, or TMZ, which is commonly used to treat glioblastoma. But here's the problem. Cancer cells are crafty. They've developed ways to resist these drugs by repairing the DNA damage before it can kill them.
That's exactly the challenge this research is trying to address. The cancer cells use what's called the G2M checkpoint in the cell cycle to pause cell division and repair the DNA damage caused by these drugs.
So it's like the cancer cells are calling a timeout to fix themselves before continuing to grow and divide?
That's a perfect analogy, Alex. And what these researchers discovered is that if you can prevent that timeout from happening, you can make the alkylating agents much more effective. They identified a specific kinase called MIT1, which plays a crucial role in this process.
Okay, so what exactly is MIT1 doing in this process?
So MIT1 is essentially the gatekeeper that prevents cells from entering mitosis. That's the phase where cells actually divide. It does this by inhibiting another protein called CDK1. When DNA is damaged, MIT1 helps keep the cell in a paused state so it has time to repair that damage.
And the researchers found that if you block MIT1 using either genetic techniques or a small molecule inhibitor called RP600-306, the cancer cells are forced to enter mitosis before they've repaired their DNA. This leads to catastrophic problems during cell division and ultimately causes the cancer cells to die.
What I found really impressive about this study is their approach. They used this comprehensive genome-wide CRISPR screening to identify MIT1 as a key target. They basically knocked out genes one by one to see which ones, when missing, would make cancer cells more sensitive to TMZ.
Yeah, that's cutting-edge stuff. And what's really exciting is that they found this synergistic effect, not just with TMZ, but with other alkylating agents too, like BCNU, busulfan, and dacarbazine. That suggests this approach could potentially work across multiple cancer types.
Exactly. And they didn't just stop at cell culture experiments. They went on to test this in mice with glioma tumors. When they combined TMZ with the MIT1 inhibitor RP6306, the mice survived significantly longer than those treated with TMZ alone.
So, to break it down for our listeners, alkylating agents like TMZ damage the DNA of cancer cells. Normally, the cancer cells would activate MIT1 to pause the cell cycle and repair that damage. But if you block MIT1 with an inhibitor like RP6306, the cells are forced to divide with damaged DNA, which causes them to die.
That's a great summary, Alex. And the researchers call this abrogation of the G2M checkpoint, basically disrupting that critical pause that cancer cells rely on. It's a clever strategy because it turns the cancer cell's own protective mechanism against them.
I'm curious about the potential clinical applications here. Do you think this could make it to human trials soon?
While the MIT2 inhibitor they used, RP6306, is already in early clinical trials for other cancer types. So it's not a huge leap to imagine combination trials with alkylating agents. What's particularly promising for brain tumors is that RP6306 has a small molecular weight and is lipid-soluble, which means it might be able to cross the blood-brain barrier.
That's a crucial point for glioblastoma treatment, since getting drugs across that barrier is a major challenge. And given that the current standard of care for glioblastoma only extends survival by a few months in many cases, any improvement could be really meaningful for patients.
Absolutely. And this approach of targeting cell cycle timing rather than directly targeting DNA repair is somewhat novel. Most efforts to enhance alkylating agents have focused on inhibiting DNA repair proteins like PARP1 or ATM.
I think that's what makes this study so interesting. It's approaching the problem from a different angle. Instead of trying to prevent the repair process, it's forcing the cells to move forward in the cell cycle before they've had a chance to complete those repairs.
Right, and the researchers showed this mechanistically through some elegant experiments. They used time-lapse microscopy to actually watch what happens to the cells. In cells treated with both TMZ and the MIT1 inhibitor, they saw the cells entering mitosis much earlier, about 5.6 hours after treatment, compared to 14.2 hours with TMZ alone.
And those cells that entered mitosis too early showed all sorts of problems: lagging chromosomes, DNA bridges, multipolar spindles, basically complete chaos during cell division that ultimately led to cell death.
It's fascinating how they confirmed this mechanism by using a CDK1 inhibitor called RO3306, which prevents cells from entering mitosis. When they added this inhibitor along with TMZ and RP6306, it actually rescued the cells from death. That really proves it's the forced entry into mitosis that's causing the cell-killing effect.
You know, this reminds me of the concept of synthetic lethality that's been so successful with PARP inhibitors in BRCA-mutated cancers. You're not directly targeting the cancer mutation, but you're exploiting a vulnerability that only exists because of that mutation or because of another treatment you're giving.
That's a great parallel, Alex, and it highlights how understanding the basic biology of cell cycle regulation can lead to new therapeutic strategies. The authors actually mentioned that their findings could potentially be applied to other DNA-damaging therapies too, like radiation.
So to wrap up, this study really demonstrates how targeting cell cycle timing through MIT1 inhibition could be an effective strategy to enhance current standard cancer therapies, potentially leading to better outcomes for patients with glioblastoma and possibly other cancers treated with alkylating agents.
Exactly, and it's a great example of how unbiased screening approaches can uncover new therapeutic targets. The researchers didn't go into this study specifically looking at MIT1. It emerged from their comprehensive genomic screen as an important player in TMZ resistance.
Well, that's all we have time for today. Thanks for joining us on Stork Science World. If you enjoyed this episode, please subscribe and share with your colleagues. And remember, this podcast is brought to you by Stork, the research productivity software, helping scientists work smarter.
And we'll be back next time with more cutting-edge research from the world of science. Until then, I'm Jamie.
And I'm Alex. Thanks for listening.